recent few years, a number of studies have discovered that long non-coding RNAs can act as sponges for RNA-binding proteins. And one such example is the relationship between NORAD and the familial proteins. So NORAD is a highly expressed conserved mammalian lncRNA, and PAM1 and PAM2 are two homologous proteins, which I call them familial. And in this example, so NORAD contains at least 15 functional binding sites, which are shown here. And when NORAD is expressed, a lot of familial molecules bind to NORAD and modulate the activity of familial. And when you knock down NORAD, a lot of these molecules, PAM molecules, become available now in the cell to bind to its own targets, and you see a hyperactive familial. And this leads to aberrant mitosis in many cell types. So our goal in this study is to discover additional such relationships from existing data sets. And to do this, for well-characterized RBPs, we define the target gene sets using clip data and also knockdown data sets. And we defined a length-matched background gene set, and the last step is to find highly expressed lncRNAs, because most of them are lowly expressed, that contain many clip peaks for the RBP of interest. And for each such lncRNA, we check whether this is a potential sponge. So we downloaded two RNA-seq data sets from Expression Atlas, and the first analysis that we have done is to calculate the Spearman correlation coefficient between NORAD and the genes in the target set and also in the length match background set. And we see a statistically significant difference between these, and the correlation with the target genes is higher. This is true for both RNA-seq data sets, and this is expected because familial proteins downregulate their targets. And this is just to check whether this is not by just random chance. We tried this for many control lncRNAs that don't contain any PAM clip peak, but are also highly expressed. I'm showing you the p-values here, and you don't see a very statistically significant p-value. Most of them are not significant. And the next analysis that we did was to predict target gene expression using two linear regression models, where in the first one, we only used the expression of the familial proteins, and in the other one, we also included NORAD expression. And to measure accuracy, we calculated the Spearman correlation coefficient between measured and predicted gene expression on held out data sets. So here the y-axis reflects that's average cross-validation Spearman correlation coefficient, and we see an increased accuracy once we include NORAD. That's shown with the orange color, and this is true for both data sets. So including NORAD improves the gene expression prediction accuracy. And lastly, I would like to go over this potential sponge relationship that we predict between TUG1, the lncRNA TUG1, and LAVAL1. So here we see a lower correlation with the target gene set, and this is expected now because LAVAL1 stabilizes its targets. So when TUG1 is, for example, the expression increases, less LAVAL1 will be present for its own targets, and its own targets will be less stabilized. And we also can replicate the linear regression result as we've seen for NORAD and PAM proteins. So including TUG1 in addition to LAVAL1 improves the prediction accuracy on this data. And since this knockdown data was present for TUG1 in lung fibroblasts, we utilized this to plot the log-fold changes after knockdown, TUG1 knockdown for targets and background. And we see that the targets are more stabilized. And this is again expected because TUG1 is depleted, LAVAL1 cannot bind to TUG1 anymore. So it will be bind to its targets, and its targets will be more stabilized. Okay, so with that, I would like to thank my graduate student, Sabar, who did an excellent work. He performed all this analysis, and please visit our poster 8277 today at 6 p.m. for more details. Thank you.
Great. One or two quick questions?
Thank you very much. I was very pleased with your talk. Some time ago, my group was working on predicting lncRNAs being a sponge of microRNAs. And it's nice to see now a similar thing with proteins. And one of the things that we worried a lot about was the number of binding sites and the spacing, the space between these binding sites, because for a sponge effect, ideally you will need to have multiple sites, but also allow sufficient space between those to allocate the bound molecules. So I was wondering, did you take that into account somehow? How are you really controlling for these aspects?
So most of the predictions that we made, the clip peaks are like more than 30. We see like more than 30 clip peaks on the lncRNA. So I don't think all of them are overlapping, but that's a good suggestion. We should count the number of binding sites on these clip peaks and whether they are separate from each other. We haven't looked into this.
Okay, maybe you'll have a look.
But we are not looking at just like 3 or 5 peaks. You know, in that case, there is some risk of overlapping, but I think that's a good suggestion. Thanks.
Another question?
I had a biology question.
Okay.
So
I'm not a biologist,
I
as
know.
you know.
But still, you hypothesize that you have the relationship between TOG1 and LL1, right? What's going on there? Do you know where is it operating? What's it doing? Why? So what's the background story about that?
We haven't looked into this. We just know that TOG1 can localize in the cytoplasm as opposed to
anything.
TOG1 is expressed in brain and retina mostly, but also in the other cell types. But we haven't explored like more.
Great. Thank you very much.